Jewett
and Mrs. Schramm,
and we're going to talk about mutations for this short online lecture. First, let's define a mutation. A mutation is any permanent change in an organism's DNA. This can happen in any cell of your body and cause something like maybe cancer, but it's only going to happen, only if it happens in your gametes is it something that can be passed down to your offspring. A mutation can be a result of just a mistake during replication, or there could be something in the outside environment that causes a mutation to occur. If there is something in the outside environment causing this mutation, then that agent in the environment is called a mutagen. On this slide, you can see that there are 5 different examples of mutations. 2 of them are fake and 3 of them are real. Can you tell which is which? Obviously, this cartoon with a 3-eyed fish is not real. That is a cartoon, and it's from The Simpsons. This frog here is also just photoshopped. However, this turtle here with 2 heads, this dog with extra muscles, and this x-ray here of someone with cystic fibrosis are all real mutations that can occur through one of these processes we're going to talk about today.
gene mutations. There's different levels of mutations we can talk about. We're going to focus right now on what we call point level mutations or point mutations. This is because we're dealing with specific nucleotides. So we're talking literally about A's, T's, G's, or C's if we're talking about a DNA mutation, or A, U, G, and C if we're talking about an RNA mutation. Either way, as Mrs. Jewett said, we have mutated what the original strand should be. So what's happening is we're taking one nucleotide and substituting it for another, or you'll see there's other things we can do here too. Just look at the difference between the normal one on the left and the mutation one on the right, and make sure you understand why we're talking about just a single nucleotide or a point level mutation.
These point-level mutations can cause a couple of different things. Nonsense, if it's not caught by one of the enzymes that does the proofreading, will cause an early stop codon. So this is going to cause the protein to be cut end prematurely and be very small and non-functional. A missense mutation is where only one amino acid will be changed. So the rest of the protein will remain the same. One amino acid is going to be different, so that might change the function of the protein just slightly and can be a beneficial change to an organism. This type of mutation is the most likely to cause a beneficial change when we talk about mutations. So here's an example. If we have this sentence, the cat ate the rat, each of those words represents one codon because it's 3 letters, just like a codon is 3 nucleotides. So if we were to cut this protein and have an early stop codon, the period comes in the middle of the sentence, making this protein non-functional. It is not giving the message that it is supposed to give. If we have a missense mutation, just one of those amino acids is going to be changed because one letter changes. It still makes sense. It's still passing on a message. The message is just slightly different. So this can cause a slight change in an organism, possibly a beneficial change.
Something else that can happen, since we know we have to follow the rules, if you remember back to translation, at the A site, we have to have 3 nucleotides make a codon. Therefore, if we start changing the number of base pairs or nucleotides within that codon, we can have something called a frame shift. A frame shift occurs in the factory at that A site when your body tries to maintain the integrity of a codon. Meaning, if for some reason we don't have the right number of codons present, your body will shift the mRNA code to make it have the 3 letters. 2 basic ways we can see this is an insertion or a deletion. An insertion just says that RNA polymerase kind of messed up, and obviously when it's decoding DNA, it puts in an extra base pair. So if it's supposed to do AUG, it accidentally does AUUG. You can see how that's going to affect the entire line of amino acids down from that as we continue to try to maintain 3 base pairs. Or RNA polymerase messes up and it's supposed to put AUG in and it just puts AU. Again, that's going to shift the entire reading frame of the entire amino acid codons so we can maintain that absolute of needing 3 nucleotides for a codon. So this is going to disrupt perhaps just one amino acid or many amino acids. It's hard to say until we look at the entire strand. Here's an example. Again, Mrs. Jewett has chosen words that are 3 letters to represent a codon. The is just like AUG, except we're using English so you understand here in this example. So this message is the cat ate the rat. That's the complete protein the way it's supposed to be. In this case, we deleted the T. Oh, we didn't. RNA polymerase did. So instead of being able to read the cat ate the rat, your amino acids are messed up. We had to shift the reading frame in the A site of the factory. So now we have heck at a tet hair at. Sure, you can understand what her means, but what do the rest of those things mean? By frame shifting the reading so that we could maintain 3 base pairs in our codon, we've completely changed this entire protein. In this case, we've inserted something. Now we have an insertion of an extra letter, an E. So the is correct, and then what happens? This insertion caused a frame shift. We've shifted everything to attempt to continue to read in 3 nucleotide codons. Again, look at the integrity of this entire message. Doesn't make any sense to me.
All right, here's just the same thing in a different format. If this is your normal DNA strand, and we insert an extra T right here, then everything from that point over is going to be different. Everything before that is the same as it should be, and everything following it will be different amino acids. The same thing happens if this one is deleted here. All of the letters are the same, they're just now grouped together differently, so they aren't being read accurately in the ribosome. All right, here's a study tip. In your notebook, describe frame shift in your own words. You might want to pause the video now in order to give yourself time to do that. Of all of the types of mutations, which do you think is going to cause the most problems? Which do you think will cause the least?
And keep in mind, before we move on just for a second, remember that when we talk about the base pairs or point level mutations dealing with just an A, U, G, or C, that if we substitute an A for a U, we are not going to frame shift because we haven't done anything to the number of base pairs in the code. If we substitute, we again haven't done anything to the number. Frame shifts are only the result if we add an extra base pair, an insertion, or remove one, a deletion. These next few slides, if you want to go ahead and look at them, please do. The other type of mutation that are large-scale mutations are called chromosomal mutations. We're not going to talk about this right now. This will be what we pick up in the spring. But just compare the size and the sort of amount of DNA involved and the differences between a single base pair, A, U, G, C, or large arms of the chromosome. So flip through these slides, but you don't have to do anything with the actual information yet.
All right, that's all for today. Thank you for joining us.
Thanks,
Enjoy
guys.
Thanksgiving.
Bye.